Hello, this is Dr. Morris. I'm doing a review for Exam 2 for microbiology. The course number is MCB 2000. This is for section .001 and section .050. Again, this is for both the lecture and the lab portion of Exam 2. I'll start with the laboratory portion of the examination. On your lab, you've been asked to review some particular media presentations, one of which is on salmonella. I would be able to recognize salmonella by the images that are shown here in front of you, though just remember each of the questions on the laboratory examination, you should be able to answer the questions from the information that's actually given in the question itself. The pictures are given just as supporting documentation, but you should be able to answer each of the questions even without the photographs that will be provided. But the image in front of you now is an image of salmonella. You're looking both at the microscopic slide to the right, and then you're also looking at just a small one picture out of the media presentation that you have seen on your laboratory examination. You should know what environment salmonella lives in. You should know the pathogenic form of the organism, where typically you can be infected from the organism, such as by the ingestion of food, exactly how the pathophysiology works, how do you achieve this infection. You should know the particular type of organism that's involved, what the particular illness is that is contracted when you ingest the salmonella virus. The laboratory that you've also evaluated in the last assignments, especially in assignment 3, was on the human immunodeficiency virus. There are a number of questions related to the human immunodeficiency virus. There is a media clip under the lab section of the virtual lab in, let's see, lab 3. It will discuss the history of HIV disease, where it evolved from over the past hundreds of years, how the HIV virus is arranged, how the nuclear arrangement of the nucleic acids are. You should be familiar with that. You should be familiar with terminology such as protein spikes and why the protein spikes that coat the HIV virus are so important. You should know what's meant by the term receptors and how certain receptors on the human cell allow the HIV virus to be more virulent, what the roles of those receptors are. You should know the enzymes that's responsible for converting the RNA of HIV into DNA. You should also know what the process of integration means as it relates to the HIV virus. You should also know the factors that strongly influence the virulence of the HIV virus. Again, the media clip is very comprehensive. I recommend that you review the media clip in Laboratory 3 if you have not already done so, and if you have done so, I would go back through and make sure that you listen to that information, because again, there are a number of questions related to the HIV virus. These other photographs that are seen here relate to the HIV virus, the receptors, the protein spikes that are a part of the virus, how the virus comes in contact with the cells, the human cells, and invade the human cells. This is again electron microscopy demonstrating the HIV virus and how it's coming in contact with the human cell and will eventually invade the human cell. The next organism that it's important to be able to identify is the Treponema pallidum. You need to know what disease is related to Treponema pallidum. You need to know the characteristics of this organism. You need to know the way this particular organism is treated in the event that it causes an infection. You need to know the mode of transmission of this particular type of organism. You need to know the length of infection that can occur with this particular organism, whether or not the body can develop a natural immunity to this organism. Treponema pallidum. Here's some other pictures. The picture on the left, again demonstrating the morphology of this organism, which you need to know. The previous picture as well demonstrated the morphology of the organism. You need to be familiar with that. The picture on the right is actually one photo out of the clip that you watched about this organism. Again, you need to know the morphology of the organism, general characteristics, the disease that it causes, how do you treat this particular organism if it causes disease, and if the body can develop any natural immunities to the organism. The other organism you need to be able to identify is Neisseria. This is a gram-negative diplococci. Gram-negative diplococci, very pathognomonic for Neisseria. You do need to know the infectious forms of Neisseria. You need to know how this organism can be transmitted. You need to know its effect on newborns. Newborns can how the newborns can develop Neisseria gonorrhea during childbirth. You need to know if there are any other diseases that can be caused by this particular microorganism. If so, you need to be familiar with those different types of disease. You do need to know the characteristics of this disease, the morphology of it, where it can exist in the environment. Neisseria, this is Neisseria gonorrhea. Neisseria meningitidis would be another organism, same family. Also, you need to again be somewhat familiar with its picture. Anytime you see gram-negative diplococci, you should always be concerned whether that's representative of Neisseria, whether it's Neisseria meningitidis or Neisseria gonorrhea. The next organism is Pseudomonas aeruginosus. You should be familiar with Pseudomonas. You should know where this is located in the environment. You should know its morphology, what its gram stain would be, why it would be particularly problematic in certain patients. And again, Pseudomonas aeruginosus. Another organism that is not pictured is Saccharomyces cerevisiae. This is also known as baker's yeast. This is also a part of your assignment. And I would review the information related to the Saccharomyces organism, particularly about fermentation, particularly as it relates to fermentation. Review that organism. And then finally, bacteriophages were also discussed as a part of your laboratory assignment. Should be familiar with bacteriophages. You need to know where you find these at in the environment. They also have industrial and economic importance, and you should be familiar with why these organisms have economic and industrial importance. Bacteriophages. Now, for the lecture portion of the examination, let's see, I believe you have 55 questions. They're all multiple choice. The review will cover Chapter 4 through 11. Chapter 12 will not be on the examination. It will be on the next exam. You do need to be familiar with the components of the bacterial cell wall, what composes the bacterial cell wall. You do need to be familiar with the process referred to as active transport. The process referred to as active transport. And I'm sorry, let me digress for a moment and go back to the first item that I discussed. When I say components of bacterial cell wall, remember there are gram-negative and gram-positive organisms. There are also mycoplasma, each of which have a little bit of differences in their cell wall, and you need to be familiar with each of those. Again, bacterial cell walls, you need to be familiar with those. Again, you need to know cell transport mechanisms such as active transport. You need to be familiar with that. You need to be familiar with the process of simple diffusion, the process of facilitated diffusion, and how each of those differ from one another. You need to know the characteristics of prokaryotic cells. If you recall, during the lecture there was a table that I gave that presented both prokaryotic and eukaryotic cells. If you are familiar with that table, you should be able to answer the question that's related to that. Talks about general characteristics, about the organelles of each, about the ribosomal constituents of each. So you should be familiar with that, how each of them reproduce. You should be familiar with the cell structure referred to as fimbriae, F-I-M-B-R-I-A-E, fimbriae. You should be familiar with that cell structure, how it's important, what its role is, especially its role in the initiation of disease. You do need to know the constituents of the plasma membrane, the constituents of the plasma membrane. Now remember, the plasma membrane differs between prokaryotes and eukaryotes, so you should know the constituents of the cell membrane as it relates to prokaryotic cells and eukaryotic cells. You need to be able to give me the definition of osmotic pressure. Osmotic pressure. I need to know the difference between simple diffusion and facilitated diffusion. Simple diffusion and facilitated diffusion. I also need to know the role of the transporter protein. One of those mechanisms uses transporter proteins, and I need to know the role of the transporter protein. I need to know which of the two organisms, prokaryotes and or eukaryotes, which one would you find mitochondria? I need to know the difference between osmotic lysis and plasmolysis. The terms are osmotic lysis and plasmolysis. Need to know the difference in those two. I need to know the roles of lysozymes inside cells, lysozymes inside these microorganisms, inside the bacterial cell, lysozymes. Let's see. I need to know the effect of a hypertonic environment, on bacterial plasma and or cell wall. The effect of a hypertonic environment, what would happen to the cell wall. Again, I do need to know the plasma membrane of mycoplasma, how it differs from the plasma membrane, for example, of typical gram-positive or gram-negative organism. You would need to be able to compare and contrast the fimbriae, as I discussed earlier, and the pili, P-I-L-I. Again, compare and contrast the fimbriae and the pili, how they differ, what the roles of each one of those two structures are. Again, you need to be able to tell me what occurs in a cell that's placed in a hypertonic solution, and then also you need to be able to tell me what would occur if a bacterial cell is placed in a hypotonic solution. You also would need to be able to tell me what a hypotonic and a hypo-PO and a hyper, H-Y-P-E-R, and hypertonic, what the difference is in those two types of solution. And again, what would occur if you place a bacterial cell inside that solution. Remember, a isotonic solution is like a 0.9 normal saline, 0.9 normal saline. So if it was higher than 0.9%, it would be considered a hypertonic solution. So you need to know what would happen if the cell is placed in that. If it's less than 0.9, say a 0.45 saline solution, it would be considered a hypotonic solution. So again, recognize what would happen to the bacterial cell wall. I need to know what the role of the glycocalyx is. glycocalyx, Glycocalyx, the glycocalyx. Need to know what the role of the glycocalyx is. I need to know the characteristics of the gram-negative cell wall versus the gram-positive cell wall. You need to be able to explain to me just briefly a brief understanding of beta oxidation. What does beta oxidation do? What particular organic substance utilizes beta oxidation for its metabolism? When I say organic structures, you remember carbohydrates, proteins, lipids. Which one of those organic compounds utilizes the process of beta oxidation? What is the role of beta oxidation? What does it accomplish? I need to know the end products of fermentation. The end products of fermentation. I need you to be able to briefly describe fermentation. I need to know about pyruvic acid. Where is pyruvic acid derived from? What is the eventual fate of pyruvic acid? Where is pyruvic acid used at within, say, a bacterial cell metabolism? And if I did not already say it, I do need to know the definition of fermentation, briefly describe the process of fermentation, and I know I said earlier the products of fermentation. I do need to know the difference between the lag phase, the stationary phase, and the log phase when we're discussing bacterial growth. Lag phase, log phase, stationary phase, and you should also include the death phase as well. I need to know the differences between a selective media, an enriched media, and a differential media. I need to know the processes that are involved in the breakdown of glucose. There are several enzymatic steps that are involved in the breakdown of glucose. Remember, glucose has to be broken down into pyruvic acid. Pyruvic acid is then utilized for carrying out the TCA, or the Krebs cycle, and then the end products of the TCA cycle are then utilized inside the cell membrane in another sequence of events. So I need to know the complete oxidation of glucose, and again the three stages, the three different processes that are involved in its oxidation. I need you to be able to define sterilization, disinfection, and sanitation. Sterilization, disinfection, and sanitation, how they differ. I need to know the term antiseptic, what antiseptics are typically used for. I need to know the definition of bacteriostatic. I need to know the definition of bactericidal, fungicidal, and germicidal. Which constituents of a typical bacterial cell are the major targets for antimicrobial activity? Which of the components of a bacterial cell are the major targets for antimicrobials? For example, some antimicrobials may target the enzymes that are a part of the metabolic activity in a particular bacterial cell. Some antimicrobial therapy may target the proteins or the nucleic acids that are part of a microorganism. So I need to know the major targets for the action of antimicrobial activity. I need to know the characteristics of an autoclave. I need to know the positive aspects of autoclaving and the limitations related to autoclaving. I need you to be able to tell me the process of pasteurization. Who first designed pasteurization? How it was first used? I need to know the bacterial effect of UV radiation. What particular aspect of the cell does UV radiation or UV light, how does that affect a bacterial cell? I need to know what's meant by the term clear zone. Clear zone. I need to know the effective concentration of an alcohol solution that's used for the purposes of antisepsis. I need to know the treatment that's used for newborns that may be exposed to Neisseria gonorrhea. What treatment is typically provided to the eyes of newborns? I need to know a little bit about a sterilization substance referred to as ethylene oxide. How it's used, what setting would you see ethylene oxide used? I need to know examples of methods that can be used to achieve sterilization. I need to know what environmental influences can a bacteria be subjected to effectively eliminate the bacteria. An example is altering the pH of a solution can eliminate certain bacteria. Altering the temperature can affect the amount of bacteria on an object. So some types of influences that can be utilized to eliminate bacteria. Should be able to compare and contrast the following agents as antimicrobial agents: cationic detergents, phenolics, PHENOLICS, iodines, alcohol, and soap. Which one are the most effective? Which is the least effective? I need you to review each of the chemical agents that can be used as disinfectants. They're used as disinfectants typically because they denature proteins. So compare and contrast phenolics, aldehydes, halogens, and alcohols. I need you to identify some of the potential purposes of genetic engineering. I need you to be able to define the term amplification. I need you to be able to define a cloning vector. I need an example of a cloning vector. I need you to be able to appropriately classify a microorganism into its appropriate domain, kingdom, phylum, class, order, family, genus, species. You need to know that order. You need to be able to tell me beginning at the major category, going to the most specific species, and from the most specific species to the major domain, and I need that in order. You need to give me the definition of a cladogram. I need to know what's meant by the term dichotomous key. I need to know the process of flow cytometry. What information is provided by flow cytometry? I need some examples of gram-positive cocci. Whether they're in clusters or whether they're in chains. Some typical examples of gram-positive cocci. I need an example of an endospore-forming bacterium. It's an example of a microorganism that I've used pretty consistently through the lectures. I need to know some general characteristics of the Helicobacter pylori organism. What environment would you find this microorganism in? I need to know the causative agent of Lyme's disease. I need to know its physical characteristics. I need to know the environment by which you would find the Legionella organism. What environment would you find the Legionella organism? What type of diseases does Legionella cause? I need to know what's meant by the term insect vector. I also need to know how a virus would be cultured in a laboratory setting. What are some of the ways that a virus could be cultured in a laboratory setting? This covers the information that will be on the exam